Well, welcome back, everybody, for our final session. And my privilege is to introduce the moderator for the last session, Luisa Valiela, who I've had the pleasure of working with for most of the 32 years she's been at EPA. I can't remember exactly when we first got. I have a couple tidbits. One thing that we have in common is that we both came from New England to California to go to Cal for grad school, and we both ended up staying here, committing our careers to San Francisco Bay. And to that matter, Luisa, if you know, is one of the EPA players that has been engaged with us for over 3 decades, and very involved and knows a lot about what goes on in EPA in a manner that helps us here. One other quick tidbit she told me, was it your first job or one of your first jobs was she was hired by Mike Connor when Mike Connor played a big role in the Boston area. So it was one of her first jobs was with Mike Connor, and she came out here before Mike came out here. So I guess Mike followed you. So come on up.
There we go. A foolish consistency is the hobgoblin of little minds. So I didn't really want to stand behind that. I kind of wanted to move around. I'm Luisa Valiela. I have been at EPA for quite a while. It is a privilege, my job, that I get to work in the water division. Is anyone surprised to see me here? There are a lot of EPAers actually here. In case you hadn't heard, EPA is still working right now. But it is a day-to-day thing. We do expect to be furloughed, but it's not quite yet. So Tom started the day by kind of shouting out, and a lot of you have, which is we run a 50 million-ish a year program for San Francisco Bay restoration, water quality, and habitat. So he was talking about the RMP and the nutrient management program getting an influx of funds, which we are really, really happy to have been able to deliver last year and this year. And he called it more and better. And we are really, really glad to do the more part. You all do the better part. You deliver that better. It also is worth acknowledging that the RMP has had a really long history without federal funding. For years, your funding model did everything. You could call it a shoestring budget, but you did it all, creating this long-term data set without federal funding. So it is about time that we did the more, and yeah, really, really happy to be here. So the next set of talks that we're going to hear are really the ounce of prevention that kind of delivers better than the amount of money that would be paid to do the cure, which we talk about a lot. Mercury, PCBs, those are all the amount of money that we have to pay now because we didn't see it coming, or it was too late when we saw it coming. So we're going to hear from Martin and Dave, Martin Trinh and Dave Senn, on nutrients and CECs in stormwater. They also will thank Rutgers, MIT, and Duke, so East Coast schools training the brains that are going to share the next two talks here on the West Coast. So we are extremely lucky to have both of them be the brains behind these two amazing programs at the RMP. So I'm going to turn it over to Martin for the first talk.
Hi, everyone. Thanks for staying throughout the whole day. I hope me and Jay's favorite turkey sandwiches didn't put you to sleep. I know they have like the tryptophan or whatever is in those, but we'll make it, not too quick. But yeah, so I just want to again thank you to LaDonna, Will, Jay, the whole APP team for sharing about our fishing work. I'm going to put on my other hat now, which is the stormwater work, which is typically about half of what I do here for the RMP. And in fact, it was a little more than half because last Monday or 2 days ago, me, Matt, Ezra, Jennifer, who's not here, Kaylee, we were all out in the storm sampling. Luckily, it was a daytime storm. So, started off the season, quite easily. We were sampling there for microplastics, which is not what I'll be sharing about today, but maybe that's a future presentation coming up. So today I'm going to share with you the RMP status and trends wet season pilot. So just to give a background on the RMP status and trends, this program began, in 1993 with dry season collections of bay water every 2 years, as well as other, mediums of like biota and sediment as well. But we started with legacy chemicals such as copper, cyanides, PCBs, and mercury. But around 2020, the emerging contaminants work group decided to ask if the RMP could add some of what back then was emerging contaminants to the program. I know Tom doesn't like that word now, but that's going to be our PFAS, bisphenols, organophosphate esters, and as a special add-on study to piggyback off this status and trends work, some tire wear chemicals. So the goals of this study were just to characterize the contaminants, these CEC concentrations during the wet season, and just increase our understanding of stormwater as a pathway for CECs into the bay. And so we sample typically after storms just to get an idea of these concentrations, develop this optimized design that we're going to continue to evolve and grow in the future. Right now we're working on stormwater, but this will all connect into the status and trends with biota, sediment, and whatnot. So for our sample collection, overall we sampled 7 storms from the fall of 2021 to about February 2024. I should have put the pictures of us at the baler, but yeah, we used a baler and a rope, not rocket science, but good science nonetheless. And so here you can see in the green, we have what we call our near field stations. So in the green triangles, if you can see, I can't really move the mouse, but that's going to be closer to our watershed. So you'll see 4 stations, one in the Central Bay, in the San Leandro Bay, and then as you go down, you'll see one in Redwood Creek, one in Palo Alto, and one in Stevens Creek. In addition to those, we had what we call our paired wise to those, were what we call our open bay sites, and those are going to be piggybacked with the USGS RV Peterson, which goes out on cruises, twice a month. The RMP and the NMS funds those cruises, but we take advantage of those cruises by also doing some of the sampling of the CECs, in addition to what the ancillary parameters that they sample as well. And that's usually done within 2 weeks of a storm. Okay. All right, and PFAS samples were analyzed by SGS Access, and a big, as we started monitoring PFAS in the past, we would only be able to analyze 13 back then, but now, since we switched to SGS Access, we're now able to have a target suite of 40 different PFAS, and maybe we'll add non-target in the future as well. Unfortunately for us, our organophosphate esters and bisphenols were also analyzed by SGS Access, but they encountered some contamination issues as well as some method detection limit issues. And so because of that, we are not going to share that data today, but certainly those will be in consideration for future iterations of this monitoring. And stormwater CECs, that should be tire wear particles and vehicle-derived particles, were analyzed by the Kolodjic lab at the University of Washington. And Ezra has a report. Chemicals, not particles, exactly. Were analyzed by the Kolodjic lab at the University of Washington, and there's going to be a different report coming out about those. So I'm going to start with our PFAS concentrations. So, I'm sure we all know what PFAS are, but for those who are unfamiliar, these are going to be these forever chemicals that have been popping up in the news. They're toxic to people and wildlife. Jay has explained that, we are particularly concerned with PFOS, which is a subanalyte that accumulates in aquatic life, bioaccumulates in aquatic life. And as you can see from this graphic that was designed by our EI team, that they're in, just a variety of different products, and that is going to leach into our stormwater runoff, and the purpose of this study was to see how much is going into the bay. So yeah, just looking at our open bay. So if you remember our sites that were closer to the middle of the bay, what we call the spine of the bay, you can see that we found 11 different PFAS. 8 of these PFAS were found in more than 60% of samples. You can see in the salmon color, we have PFHXA. Some people call it PFEXA, which I may or may not use throughout this presentation. We have PFOA and PFOS, which we're familiar with. PFOA in the red and PFOS in the blue. And as you can see from these profiles, that these 3 comprise the majority of these PFAS profiles. But yeah, you can see they are organized by site, and temporally we don't have enough data to make any associations or conclusions yet. Next, going to the near field, these are going to be our stations closer to the watershed. We found 16 different PFAS, which is more than the 11 we found on the open bay. 9 of these PFAS were found in more than 60% of samples, 8 of which being the same as the ones in the deep bay, plus PFDSA. Again, PFHXA, PFOA, and PFOS were all found at the highest levels and comprise the majority of the PFAS profiles. And again, I just want to note this different scale. The last one I think topped out at 30, and this one is right about 140. So quite a considerably a larger amount of PFAS found in near field samples, which you'll see in the next slide as well. So here on the left, you can see our near field stations. On the right, our in-bay stations, and they're color-coded by their pairwise associated part site. And you can see that, again, this concentrations in the near field are much higher than those in the open bay. You'll notice that we observe no real pattern in our near field sites, but in the open bay sites, you'll see that as we go from left to right, that's going to be north to south. So starting from the central bay, working down to the lower south bay, you'll see concentrations gradually increase, but that's due to different hydrodynamic processes. Our central bay site is like deep out in the middle of the water versus the hydrodynamic processes in the smaller lower south bay are just going to lead to less dilution. In our near field sites, you'll find that PFAS and subanalytes such as PFOS, PFOA, and PFEXA follow the same trend or lack of trend by station. We have higher concentrations in the central bay and in the lowest south bay, but our middle 2 stations in the south bay and lower south bay, had slightly lower levels. And there were no significant differences in PFAS levels for these near field stations. For the open bay, again, we notice we're gonna have increasing concentrations as we move from the central bay to the lower south bay. And again, these sites are gonna follow the same pattern for some PFAS, PFOS, PFOA, PFHXA. And so we also took samples in the dry season to compare to our wet season samples. And I have a slightly confusing graph, but I'll attempt to walk you through it. So on the left, we're going to be comparing near field samples in the dry season to open bay samples in the dry season. Near field in the red and open bay in the blue. And you'll see that again in the same pattern as the wet season, we observed that near field concentrations are going to be higher than those in the open bay. But we had slightly more interesting results when we compared to within just the dry seasons, or within between the dry season and the wet season. So on the right 2 box and whisker plots, you'll notice that we're comparing the wet season directly to the dry season, wet season being in the lighter blue and dry season being in the darker blue. And there you'll see that the wet season samples had slightly higher PFAS concentrations than our dry season samples, but not significantly so. And this could just be, a testament to the forever aspect of our forever chemicals, as well as different contributions from wastewater, as well as different hydrodynamic processes. And again, you'll see that illustrated in comparing the wet season to the dry season, in both the near field and the subanalytes there, and the open bay. And so for salinity, a key part of this study was making sure that we took samples at the correct timing to ensure proper mixing, making sure that we were capturing runoff and not just bay water. So, we would expect that, the saltier bay water would have less PFAS than the fresh water in the watersheds, the runoff from, both stormwater and wastewater. And that is what we found. We can see that that is the case. So we typically try to get concentrations, time our sampling, so to be between 15 parts per thousand to 20 parts per thousand of salinity, which is, I think bay water is 30 parts per thousand of salinity. So we were trying to get a well-mixed bay. Unfortunately, our timing was not great. And so we only got 3 out of 20 samples in that range. But we do notice this pretty clear correlation that as you increase in salinity, your total PFAS concentration is going to decrease. And again, that's just due to dilution and other factors as well. And yeah, you'll notice that on the left, we'll have salinity, and on the right, we'll have total PFAS concentrations for our sites with low salinities, which is going to be San Leandro Bay and Stevens Creek, which were quite deeper into the watershed. We notice higher PFAS concentrations, whereas our sites, Redwood Creek and Palo Alto, which had higher salinities, had lower PFAS concentrations. So key takeaways from our PFAS data are that concentrations increase as you move south from the open bay, with Central Bay concentrations being significantly lower than our lower South Bay concentrations. We didn't observe the same effects for the near field sites, and again, that's just due to the different hydrodynamic processes in those particular watersheds. And we noticed surprisingly that concentrations were similar in both the wet and the dry season. And again, we just realized how important salinity is, and properly timing our samples is to making to our PFAS concentrations. So for other CECs, this part is going to be a little less exciting because, again, we had these field blankets for our organophosphate esters and high method detection limits in our bisphenol. So that data, we're still scrutinizing that data, but we'll look to improving our processes as well as our analytical processes in future iterations of this sampling. As well as for tire and vehicle-derived contaminants, that report is currently under draft by Ezra. They did notice, or preliminary findings indicate that there are detectable concentrations of these contaminants in the open bay and the near field, but these are below available toxicity thresholds. Yeah, so be on the lookout for that report, probably after baby. No, before. There we go. All right. So yeah, going forward, we're convening a small group to work to make some recommendations going forward for our program. We're gonna get started, and hopefully, we didn't think that the water year would hit us so quick, but here we are, and hopefully we're blessed with a lot of water and a lot of samples this year. The pilot study, generated this small initial data set of 7 storms for 4 near field sites and 4 open bay sites, but we need more data than that to draw any real conclusions. Again, data have not been generated for organophosphate esters and bisphenols, but which may behave differently from PFAS, so that's definitely crucial data that we'll need going forward. We definitely are considering the ideas of including different sites. We haven't hit the North Bay yet. That's something that we're definitely going to talk about tomorrow at our small group meeting, and really pondering this question of timing and salinity at the sites that we've selected. And all of this feeds into different aspects that the RMP is working on. So for example, this tracer study report coming out from Ali King and the modeling team really try to quantify the relationship between the near field site of Stevens Creek to our open bay site of, we call it 34, but that's just our lower South Bay site. And it found that, these conservative tracers found that there may be not as much associations between that near Stevens Creek and site 34 as we thought. But all of this go hand in hand to inform, including the upcoming NMS work that Dave will share with you about coming up. And coming soon is this report, which will, again, we're convening the small group. We're going to hope to finalize that draft report and have it out to the larger RMP soon. Again, I just want to thank the Stormwater team, everyone else who has worked on this stats and trends work, as well as, even our intern Jonah, who is the face of a couple of these pictures and now is was in Antarctica and is doing lots of different things. But lots of different people have come to help with this sampling and with this work. Yeah, and please reach out if you have any questions or ideas.
Are we going to do one question for Martin, or are we going to wait till the panel? I don't know about our timing. Yeah, Martin, I think you're going to get a question, Mr. Stormwater.
Hi, Martin. Yeah, cool stuff. So one thing I noticed was sort of like the San Leandro Creek and Stevens, right? They had a lot more variance in concentrations, as well as kind of occasionally going higher. So part of me wonders if we had better control on salinity, like if we waited long enough for both those places to get higher salinity, would they have looked like the other sites more or not? And that we can't know. But
yeah, that we can't know. I think it's certainly a possibility. Those sites typically had, I think, lower salinity, so indicating that we either waited too long or we didn't go fast enough. But the thing is, we have small crews and not enough personnel, which may be increasing due to this upcoming money, but hopefully we'll have the capacity to really, target storms in the way that we want to be able to. Or, an upcoming plug for the remote samplers is that they would be able to find the salinities that we want and sample exactly when we want, as opposed to having, crews just go and target all these sites all at once.
So in your salinity plot there was that one like freshwater sample more or less that had like no PFAS in it. Any ideas what was going on with that one? Or did you find a mythical source or something?
I could not tell you. We're going to dig deeper into that. But yeah, we'll see.
Cool.
Thanks, you were just here. Sorry about that. So there was one slide that I was looking at water years. I know you talked about wet season versus dry season. The wet season typically had higher concentrations of contaminants. Did water years seem to influence that level of concentration that you had across years? Like there's a plot where you show like 2021 to 2024, I believe.
Mm-hmm. We haven't taken that granular of a look at the data yet, but again, we only have 2 storms per year, so I don't think that at this moment it's not representative enough of what's going on. And sometimes we were able to get first flushes, other times we had competing studies that needed to be sampled. So, all right. My time's up.
All right, our next speaker is Dave Senn, who leads the nutrient management program at SFEI. He has built out an amazing team, which I'm sure he is going to thank, but we are really, really blessed to have lead brain, Dave Senn.
Thanks, Luisa, for the introduction, and glad to see everybody today. Yeah, my name is the only name on here, but this is definitely the work of many, and I'll be talking about some of those many as we go through this presentation. So the overview of today's talk, or the title of today's talk, Science to Inform Management, the NMS Science Priorities for the next 4 years, related in this recently issued permit. So as a background graphic, just San Francisco Bay. San Francisco Bay is highly enriched in nitrogen. Sorry, I forgot my reading glasses and I can't see my own notes. Maybe if I step back here. Is that right? No, I'm good. I'm good. Thanks. That's okay. No, that's okay. I'm just. That was just an excuse to get over the nervous stutter. That's all. So highly enriched in nitrogen from wastewater treatment plants as the primary source, relatively resistant to that nutrient-rich status, but nonetheless at risk of impacts should any of the conditions that keep things in check be lifted. So historically, phytoplankton growth was primarily light limited by strong tides and high turbidity, and we are more or less on the left-hand side of this plot. Like high nutrients, but maintaining low or moderate production, ample oxygen, limited to primarily spring phytoplankton blooms that were considered pretty good for food supply, and rare HAB flare-ups. Plenty of HAB organisms at low abundance, but rare flare-ups. And that's been held back by the concern has been, as we've seen over the last 15 years, evidence consistent with changing dynamics or responsiveness or sensitivity of the system, that there would be a potential for adverse impacts to occur with the same nutrient levels present in the system. So essentially, where we would shift from condition A to condition B, either on an ongoing basis or more frequently and episodically. So large blooms, low oxygen, and severe HAB events. So the summer of 2022 severe harmful algae bloom of this organism, Heterosigma, it's no longer a question of if or whether this could ever happen, where that dam could drop down and conditions could worsen. We've now seen it. And in fact, it was arguably in some ways the worst case scenario, in that all of the nitrogen that was produced or present between, let's say, the Bay Bridge and the Dumbarton Bridge was utilized by phytoplankton towards ill means. And so we actually not only did that bridge drop down a little bit, or that barrier drop down a little bit, it dropped down all the way. So going forward, one of the key things we need to be thinking about is how frequently is the bay going to be able to remain mostly in that condition A, or is it going to be flip-flopping between condition B with some frequency that would be unacceptable? So the Regional Water Board in October 2024 issued an update to the Baywide nutrient permit, and in that, they're requiring reduced POTW loads by 40%. So the permit requires 40% Baywide. It's a bit open or flexible in terms of exactly how that 40% is achieved. Nonetheless, the individual POTWs, there are limits for individual POTWs included in there. And the compliance schedule in the permit, 40% reduction within 10 years, but there's ongoing discussions about ways to potentially extend that compliance schedule. The 2024 permit notes that further reductions may be required in the future, further nutrient loads reductions in the future. One clear example that's called out in the permit is the potential for understanding what nutrient load reductions might be needed to prevent the direct lethality from the HAB event that occurred in 2022. In 2022, there were maybe 2 waves of impacts on the environment. The first was that fish started dying well before dissolved oxygen started dropping. And then a second where fish mortality increased again as dissolved oxygen levels decreased. So a reasonable hypothesis for that first wave was that it was more of a direct effect from Heterosigma and potentially would require a different threshold. Beyond just Heterosigma, there was also the permit notes other potential critical conditions. So the NMS science priorities for the coming 5 years, or 4 to 5 years, are target the information needs related to the permit requirements. Science planning process, this is our third science plan that we've developed. The first was from 2014 to 2018, 2019. The next was 2019 to 2024, and then now this updated one. The work on this, we got started on this in early January, or in 2024, early 2024, but the main effort really got underway September through 2024 through May 2025. Monthly or bi-monthly meetings with the NMS steering committee and subcommittees, and also smaller groups at various times. And I wanted to, before moving any further, there were a large number of people who invested a large amount of time and a large amount of patience into working with us through that process of developing this plan, revising, reiterating, et cetera. And we also needed to develop this plan for 2 different potential funding levels because of uncertainty on the funding side. So the 4-year science plan, we have that as our guideposts, and we revisit that annually to develop annual program plans to keep moving the work forward. So I'm going to go through not an exhaustive list of the priorities, but I'm going to go through some high points, main elements of the priorities, and then use the remaining time to talk through a couple examples of the why and the direction that we're going with those. So in general, the priorities were expanding a lot of things, expanding monitoring, expanding modeling, ample support for synthesis and data interpretation, digging in and pursuing mechanistic HAB studies, which is something that we haven't had the resources for before, as well as expanded science communication to get the information out from the work that we're doing, the technical work, out to the decision makers' hands and in a form that that's useful for them. The breakdown here of the funding, roughly 35% monitoring, 30% modeling, about 20% projects and special studies. Included in that is assessment framework, synthesis, and field and lab studies, and then 15% program management. The total budget that we planned for was a number of roughly $4.5 million, and that's roughly double what we've had in prior years. In the prior years, we had somewhere between $2.2 and $2.5 million, a combination of funds from the annual permit fees from the wastewater agencies plus funding from the RMP. This additional influx of funding came from a large grant from the EPA, which at different times was at different levels of confidence and certainty. So the major focus areas, and these are just some examples, and I'm going to come back and close with this slide and also touch on some things that I hope we're able to still pursue that aren't on this list here. So obviously, for good reason, an increased focus on HABs, expanded monitoring, improved mechanistic understanding through targeted studies, as well as analysis and interpretation of field data. And also this organism that caused the major event, Heterosigma, that's going to be a major focus. One of the things that we also need to be thinking about is that organism came out of nowhere. I mean, it was on our list to watch, but it wasn't necessarily the highest priority. And so how do we find the right balance between not looking backwards at last year's surprise problem and continuing to keep our eye on, let's say, the types of problems? This is going to be we're going to need more information, not on Heterosigma, but also trying to be able to still go broader than that. There's also a major investment in critical condition investigations, where the main focus has been thus far, or looking forward so far, is summer months with HABs is the major focus. And this is a major shift in the approach that we were taking previously. Previously, everything was about the spring bloom, excessive spring blooms and concerns about excessive growth during that time of the year. This is during a much different time of the year and a much different set of mechanisms. So one of the questions that I'll come back to the questions later. So monitoring major focus areas, harmful algal blooms, of course, data collection to improve calibration of the numerical models, as well as detecting changes, monitoring or putting the initial work in to be able to detect changes from the eventual upgrades and the responses from those, in terms of decreased nutrients or changes in other responses. And then on the numerical modeling side, the major focus areas, developing models for evaluating environmental change scenarios and HABs, as well as applying those to be able to help inform potential management decisions or potential load reductions that would protect against those scenarios occurring. That also is a departure from our work previously, and it's going to be a challenging set of issues, but I believe the team is up for the task. And then ongoing measurements of biogeochemical, of the biogeochemical model to calibrate improvements to the biogeochemical model for nitrogen cycling, dissolved oxygen, phytoplankton productivity, and also applications. And so the two areas that I'm going to talk through now are first, an overview of where we're going with the monitoring work and what we've been moving forward, And second, a bit of a focus on some of the work that we're continuing to do on improved understanding of nitrogen cycling. So on the observational data and analysis approaches, this is an area where the NMS prior to 2022, 2023 was already making major investments in expanding and really forward-thinking ways of monitoring. We're continuing to build along and continuing to work with the USGS, continuing that long-term record, but on top of that platform, we're taking really good advantage of that platform to augment the measurements that are carried out through that infrastructure or that monitoring infrastructure. One of the things in particular we wanted to point out is the additional moving away from microscopy for detecting phytoplankton and quantifying phytoplankton in HABs and towards molecular techniques that we think are much more accurate or have the ability to be much more accurate and more sensitive at detecting organisms of concern. Continuous water quality moorings. So we started back in 2013 with some initial funding from the RMP to add 1 or 2 sensors in far South Bay with the idea that we would grow from the south to the north. And that's exactly what we've been doing with NMS funding as well as with some external grants. But the red dots here with the circles in the middle, I wanted to just show you how far this network has expanded. From 2020, beginning in 2020, all the circles that have the dots in the middle, those have all been added since roughly 2020. This is an area that Ari Chelsky has been leading a lot and with a great team moving that work forward. In addition, we've been working with our colleagues at the USGS. The deep channel stations, those green squares, focus exclusively in the deepest part of the bay and don't provide information about the areas that are the large areas that are outside of that zone. And so we've been working with our colleagues at USGS to do these high-speed mapping surveys that go out and go from shallow to deep. And we've been doing those 6 times a year for the last several years. We also recommended to the steering committee that we start adding those in the northern part of the bay. And so we're now doing that as well 3 times a year in the northern part of the bay. Remote sensing, we've been using the data that we've been collecting through a lot of these activities and use that to calibrate a chlorophyll model using remote daily satellite flyovers by the Sentinel-3 satellite. It has roughly 300-meter resolution, daily information. And we now have a set of scripts that run this daily. And so we have automatic updates and a web portal for people to access the information. Data analysis and synthesis. I'm not sure if that graphic is clear to anybody. I couldn't come up with a great graphic for synthesis. So this is just stitching it all together. Those are stitches. Numerical modeling, and that's something that I've talked about a number of times here, but moving that work, continuing to move that work forward. We have a great team at SFEI and also external collaborators that feel like essentially part of the team or are part of the team, just with a different label. But this table here is showing the group and all the things that they're focusing on across all of these areas. I'll be sharing, I won't be able to go down this whole list here, but I think a number of these folks are here today, and I'm sure hopefully you've had a chance to already speak with them. One of the things I wanted to point out here as well is that we've been steadily moving towards making these data available in forms that are accessible to the public and stakeholders. I wanted to thank my colleague Dan Killam for taking the lead on most of this work. And for example, POTW loads on the upper left, trends in water quality, telemeter data from moorings, et cetera. So the next thing I'm going to go on to is to talk a little bit about the nitrogen loads, transport, and cycling, the key questions related to that. So a quantitative understanding of dissolved inorganic nitrogen loads and transport transformations is a high priority for informing management decisions. It's needed in order to reliably predict the DIN concentrations at the beginning of an event. So I'm not exactly sure what the definition of an event is, but it sounds like sort of a not every day. So let's say an event is a week or a month out of the year. We need this is the DIN concentrations. It's all the buildup for the party that's about to happen when that event occurs. And so we are aiming to try to have the best possible predictions of the DIN concentrations in advance of an event. Because the DIN concentrations at the start of the event ends up being the fuel that can make the bloom worse or less worse. So what types of data or information are needed to inform that understanding? What will be the effects of POTW upgrades on nutrient concentrations in the system? And also based on sources, zones of influence, what management actions are feasible to actually achieve certain outcomes? So again, I'm showing these loads. These are the locations of a number of the wastewater treatment plants coming into the bay. How does DIN added via these sources move throughout the system? What transformations occur? What regulates the magnitude of these transformations? What are the combined effects of transport and transformations to affect the concentrations in the bay as well as export out the Golden Gate? So just as an example of that, this is showing time series from 2005 to 2020 of nitrogen concentrations at 3 stations in South Bay. And you can see the roughly 3-fold variability seasonally, roughly 2 to 3-fold variability spatially, and also lots of interannual variability. So what are the factors that are driving these transformations? Some of the things that we're exploring here and looking at, on the bottom, I have a little description here. What are the major processes that influence DIN concentration? So it's uptake by phytoplankton as they're growing. They need it to build cellular mass. Mineralization and recycling. As organic matter dies or as there's respiration, that nitrogen comes back. Denitrification is the only true loss mechanism for nitrogen in a system. It's essentially anaerobic respiration, breathing without oxygen, using nitrate as the source, and turning it into nitrogen gas, and then burial. And this graphic here on the bottom, on the left-hand side, is showing results from an older model simulation of the loads to the system in red. The blue is showing exports from the bay, and the yellow showing losses inside the bay. And so at certain times, losses are as much as 50, 60, 70% of the influx to the bay. The right-hand side is showing a mass balance at the sub-embayment scales by month. So I want to walk you through a bit of quick data. I'm going to try to go quickly through some of this because I see the card is flashing. This is showing some nitrate concentrations sensors we deployed at the Dumbarton Bridge. You can see again this really strong variability. The gray lines are showing the 15-minute data. Red is showing daily average, but high variability. Also, we have another station just a bit to the north of the San Mateo Bridge, nitrate concentrations at the shoal mooring. And we started doing these mapping surveys with a fair bit of regularity in 2021 and noticing these really strong variations in nitrate concentrations, and also much lower than we've expected in prior years. So down around the 5 micromolar is getting to be detection limits and is much lower than what we expected during summer months in the system. We did the same thing in 2022, 23, 24, 25, and I just want to walk you through what some of that looks like here. Here's 2021 concentrations by month. Here's 2022. 2022 concentrations look comparable to 2021. 2020, sorry, that was 2023. 2022, nitrogen concentrations got off to a lower start, so maybe they were lower by the time we got into the bloom period. But in fact, it was quite a different phenomenon. The nitrogen concentrations actually ticked upwards during that time period and were peaked again, more or less, right before the bloom started. And then 2023, 2024, so we have this really strong interannual variability and between months. And the magnitudes of these changes, I'll try to go through this just quickly here, but the changes in nitrogen to go from March 2021 to May 2021 is roughly 6,000 kilograms per day. Add to that the loads that are still coming into the system, it's more like 20,000 kilograms per day. So there are really substantial losses. That's roughly comparable to the loads that are coming in from all the wastewater treatment plants at once into that part of the system. So DIN loads within this region, 20,000 kilograms per day. These losses correspond to roughly 20 to 25,000 kilograms per day from the system. The losses that I'm showing here, adding the loads plus the changes in mass from the concentration changes. And so some of the key questions we're exploring here is the variations in the biogeochemical rates and processes. We think that those are the key driver to the differences in the nutrient cycling. So in other words, algae and bacteria are performing a $10 billion activity essentially sometimes and unreliably. In other words, I use that number $10 billion. That's on the order of the cost of reducing nutrients by about 80% in this region of the bay. And so if biology is not acting the same way, we're going to end up with more or less nitrogen in the system. One of the key ideas that we think is happening here or causes is that denitrification in the sediments in the bay is carbon limited. And in other words, you need primary production to first produce the carbon that makes the nitrogen go away. But one of the downsides of that is that the more primary production that you have to produce that carbon, that's actually taking the system in a direction that we don't want it to go. We don't want the system to go in a direction of more production. We want it to be stabilizing at lower levels. I think my time is probably up. So acknowledgements, NMS Steering Committee, Planning Subcommittee. Okay. Great, well, how far back do I go? Okay, so, thank you. What sources contribute most to DIN along these shallow shoals? And what were the changes that we're seeing here? Are they because of load changes? One of the things that was really interesting to find out was that when we were seeing these very low concentrations in 2021, we gave folks at San Jose a call and found out that they were doing some tuning to their wastewater treatment plant to actually decrease the loads coming out of the plant. That was to the tune of roughly a decrease of 2,000 kilograms per day compared to their usual. So nothing compared to these load reduction, these changes that we're seeing here. So it can't be load reductions alone. But one of the things that we've been pursuing is approaches to be able to estimate where the nitrogen is coming from and the zones of influence of wastewater treatment plants so that we know the path of the nitrogen and so we can understand the processes better. And so the idea of this approach for developing or estimating the zones of influence for each of the wastewater treatment plants is to develop efficient tools for estimating the individual contributions from those plants, using that information, source apportionment, to be able to inform management decisions. We're using that information as well to identify where to monitor to maximize the likelihood of being able to see an effect from the upgrades, and improved understanding of nitrogen cycling. And so the approach here, it's primarily, at this point, a hydrodynamic model, and then adding various numerical tracers, conservative and reactive tracers, where we're using loss rates that are comparable to the loss rates that we expect based on past measurements from output from models. And then using that to look to predict the zone of influence of each of the individual sources. So this is showing all of the concentrations from all the POTWs going from very low concentrations. It's a very expanded log scale axis, so 0.1 micromolar up to roughly 70 micromolar. This is then showing the incremental DIN contributions of the individual DIN sources to the bay. And so varying from very small to very large, and you can see that there is a fair bit of dispersion or mixing from the system, but there are also clear zonations. It's not as though this is one big bathtub where you can think of everything is entirely well mixed, and that trading with one entity is the same as trading with another. This is showing the relative contributions, or the percent of the individual sources to total DIN. And so you can see here where are the dominant sources contributing to the nitrogen. One of the things that's, I believe, on the upper right, you can see the influx of nitrogen coming from the delta into the San Francisco Bay. If we take a quick look, going back to the South Bay and South Bay Shoal, what are the sources that are contributing most to the South Bay Shoal, for example? And it's an interesting mix. It's not EBDA, for example, which is by magnitude and its proximity to the shoal, you might think is having a really major influence mixing downwards. The model predictions suggest that this is actually nitrogen coming from various other sources and being shunted out onto the shoal and having an effect out in that region. One of the reasons for that is what I'm showing here is a plot of a residual circulation plot. Essentially, remove the tides from a model where the water's going like this. Well, the water's going like this, but it's also moving upwards, like a residual pattern. And so I'm not sure if you can see it very well there, but this is showing the residual circulation of the water carrying in it the solutes. And so, in other words, carrying some of the nitrogen out onto the shoals. Just as a quick application or an example, one of the things that we're hoping to be able to use a tool like this for is investigating load changes and the effects on concentrations. And so the example that we used here is load reductions. We know that load reductions from the Sacramento wastewater treatment plant came online in 2022. This is 2021. What do we think the distribution of concentrations or the change in concentrations would have been from an upgraded wastewater treatment plant in Sacramento throughout the bay? This is showing the contributions to DIN pre-upgrade. This is showing the concentrations post-upgrade. And then the panel to the upper right, or sorry, to the right shows the change in concentration. So a change of about 5 to 10 micromolar in certain locations, roughly 20–25% of the concentrations in Sassoon Bay. So I'll stop there and end again with the acknowledgements, and thanks for the mercy.
Yep, you're the panel, two-person panel.
We can have also questions for previous presenters as well.
I gotta say, I'm missing Mike Connor right now. He would have geeked out on those slides, Dave. Wow. Yeah, questions for both Martin and Dave, and there, I mean, there's a lot of things to touch on from other presenters earlier. I would even start with maybe a question prompted by Sierra and the throwing out the challenge of what's the story you want to tell through your new ways of communicating, whether it be social media or otherwise. In short bullets, Martin and Dave, what are you doing and finding in your data and in your work that you would love to share with people?
Yeah, for me, I think that this is just the beginning. This pilot study was just the beginning of a big concentrated effort that the RMP will continue to be able to expand on and grow and evolve. I think, Tom doesn't like emerging contaminants, but to us, a lot of the sampling is new and, we're not sure what it looks like. A lot of these concentrations, we don't know what this data looks like yet. So being able to tackle this problem with the addition of remote samplers, with increased staff, with more sites, I think we're all extremely excited about that possibility.
I think one of the major points that we've been moving our program towards, or motivated by, what's been motivating the movement of our program, is that much of the information we have about the behavior and the dynamics of San Francisco Bay is based on measurements in a particular area or region of the system. And it seems very likely that at least during certain time periods that it's actually processes that are happening distant from there, out in the shallow areas of the bay, that might ultimately, by circling the water back around, be the places that are shaping the chemistry in those parts of the system, that we need to be going to those places where the reactions are happening in order to understand the conditions throughout the system. And so we've been really fortunate to have, well, I would say, progressive or proactive steering committee and stakeholders who have been interested in trying to move and augment the monitoring in that direction. The second point, I guess, would be in terms of the scale that we need, that we're starting to look at things. There's the time scales, where primarily most of our understanding was based on monthly or biweekly data. We're now moving towards having data at the 15-minute resolution that we think can give us additional important information. And then spatial scales, the measurements at these individual locations where we have moorings, but also bay-wide with the long-term surveys from the USGS, and at the level of remote sensing to understand things at the whole bay scale. And lastly, that we're even trying to go for the total other direction and understand things at the DNA-based level. So, for example, our viruses, marine viruses, playing an important role regulating phytoplankton communities and HAB events in the bay, and that we may need to be looking that deeply if we want to understand certain causes.
What questions do you all have? Raise your hands, Jim. Someone want the mic?
I want to follow up on Luisa's question, which is, how do you tell the story? I've been working a little bit with shark stewards in Aquatic Park in Berkeley, Aquatic Park Lagoon, and the Lake Merritt Conservancy. And these are people who feel very passionately about the death of fish and sharks in their backyards. And they want a story as to how do we get to a better place. And as much as I get confused and amazed by David's slides, that's not a story. It's, we don't know yet. And I don't know that that's a satisfactory answer. I think people are going to want to know, when do we know? And I suspect that what killed fish in Lake Merritt and Aquatic Park is different than what killed. It's just to enlighten you that at some point you have to be able to tell a story to be able to justify billions and engage the people who are very passionate about not having that happen anymore, that you're going to need to get that money.
Yeah, we may have Sierra join the panel. Other questions, though?
So this is a question to Dave, and then maybe some of the POTW operators. You were mentioning sort of like getting down to like 15-minute or hourly scales of kind of understanding processes. And then sort of was simultaneously thinking, well, can we even manage those things? Like even if we got perfect at predicting those, how do we react to those, or how do we, do things like that? And so reducing DIN from the plants or whatever, can they do that seasonally or on a day? Like, this is a bad day, I have to really amp it up. So I have to plead ignorance on that, and somebody maybe can answer.
Yeah, Don, that's a good point. I mean, I think there's monitoring condition of a system and knowing what the steady state is, or the pseudo steady state of what the monthly averages, et cetera. I mean, that does seem like the right level to be looking at many things like condition. But I think that in order to be able to make inferences about the underlying processes, so we can report out on concentrations at high resolution or lower resolution. To Jim's point about telling a story, I think the story is going to come, like the why is going to come from, at least in some cases, from those higher resolution information that allow us to look between the time periods when we haven't had information before. Not exclusively, but I think that I think to go the next distance, that I think that it seems like we need to go.
I see Lorian's hand up, but I'm going to take two questions that we have online first, and then.
Thank you. Yeah, so these are both nutrients-related questions. Sorry, Dave, keeping you on the spot. The first is regarding the August 2022 event. So is the hypothesis for the high DIN concentrations in August 2022 that denitrification turned off during that month due to an uncanny low availability of bioavailable organic carbon? And if so, what is your hypothesis for that low carbon?
So I think if we look at those graphics again, we'll see that things shift back and forth, plus or minus a few weeks. It could have very well been that there was, let's say, a bloom that occurred earlier in the summer, earlier in the spring, was utilizing a lot of the nitrogen during that time period, and that, let's say, higher suspended sediments were present during the month of July and August. I don't have that information at my fingertips, but I think that would be an example where primary production slowed down. There was less fuel for the organic carbon. So I think that it's one of the things that we're steadily looking into, and it's one of the reasons that we're working on tuning the biogeochemical model to try to predict those things better.
Thank you. And then the second question, in a similar vein, is it correct that your estimates of DIN load from the high-speed mapping, was that nitrate only? Would changes in ammonium and nitrification of ammonium help contribute or explain lower or higher nitrate concentrations in the maps in different years?
Yeah, that's a great point, and I glossed over that. Thanks for whoever asked that question for that clarification. We did measure we did measure ammonium concentrations, sometimes underway at high frequency, sometimes by doing many discrete samples to measure ammonium concentrations. And in general, the ammonium concentrations were also low when nitrate concentrations were low. So it's not as though it was just a switch in the form of nitrogen. I don't want to say that they were zero. We wouldn't also say that nitrate was zero, but they were also low.
I wanted to respond to, I guess, the second part of Don's question. You called out if there was any wastewater operators in here who could answer about nitrogen management on a short scale, and I was waiting for an actual wastewater manager to answer, but since none of them wanted to, I did want to get back to that. So one thing I'll defer to Dave, but the nitrogen that's in the bay isn't the nitrogen that got discharged 10 minutes ago. It's been in the bay for weeks. So if we could all send our discharges to space or something, that actually wouldn't solve the problem today. And we actually did look into the question of whether agencies can hold their discharge. A handful of them actually do have that kind of storage capacity, but most of them, as one plant operator, she looked at me when I asked and she said, "Lorian, we can hold it if our community members can hold it." So unfortunately, that doesn't look like it's a great solution. It's going to be a question of longer sustained nutrient reduction efforts that we're committing to. And Jim, in terms of the storytelling, I was sorry not to see a big pile of them outside, or maybe I missed it, but the science program, the nutrient science program, put together a state of the science summary, sort of a brochure magazine type item, and it is excellent, and I definitely would refer many of you who would like to learn a little more about the science to check it out. It is available on SFEI's website.
Other hands? Tom, okay. Tom and then Tom.
So one thing, Laura, you took the wind out of one point I wanted to make, because I was going to ask Dave to speak up in terms of the substantial step improvements in communication, aka you're making some user-friendly reports available. So there's a lot of attention by the science program to that, and it's realizing fruit from that effort. So yeah, so a lot of progress there, and I think access to information is readily available through the website, because there's a whole bunch of stuff you and your team are doing on a regular basis. You did mention, as part of the use of being able to do more with more resources, be able to put more effort towards synthesis, and an outgrowth of that, of course, would be synthesis for scientists versus synthesis for managers and other parties. So basically, you don't have to follow up too much on that. One thing you didn't answer this Don's second part question, Lorian, is like the wastewater community is focusing in on dry season load reductions, and so implementing technologies that can be deployed during part of the year when there's capacity available compared to the wet season. So that's already baked into how what the permit requires and how the wastewater facilities are responding. Can't to go to less frequent changes, as you point out, is really difficult. So Martin, I just not much a question, but perhaps maybe I can formulate a question out of it. But I just want to sort of put a slightly different take on what you presented in that some historical perspective. Having been there from day one, we at the beginning of the RMP, we actually used to do surveys water 3 times a year. And then when we did one review, we decided we can cut back to 2. So we were doing wet season, dry season. And then subsequent review, we decided we could get by with just dry season. Now, part of all this was trying to find enough resources to do other things rather than just recurring status and trends monitoring. And a big driver for our ability to decide to go to just dry was copper. It was the major pollutant of interest back in the early days of the Regional Monitoring Program. And though we knew a lot of copper was coming into the bay through runoff, we observed that levels in the bay the open bay weren't substantially different between the wet season and dry season. So we could afford to keep pursuing knowledge by just doing part of the year. A lot of that had to do with being frugal with limited resources to do not enough stuff. So now that we're in ability to do more, this started before we had the more resources, because you know that we did do a review with the Status and Trends program going back 4 years now, maybe, and we had time has come to revisit that wet season question because of the growing interest in making sure we adequately are accounting for the presence of emerging contaminants in the system. So that was sort of the genesis for the pilot that you communicated on. But that pilot was also constrained by limited resources. Like how much could we afford to do? So we had a limited number of stations focusing just on the South Bay. So I know we're going to have this dialogue in terms of where do we go with the wet season monitoring. It's a given that there's a benefit to continue to do the wet season monitoring, but I don't expect you to have a thorough answer, but I think if you had your druthers, I would suggest that you tell us about being able to do more stations more often during the wet season to get a better handle on representative monitoring that would make a difference.
Yeah, just echoing everything that Tom said. Again, we kind of noticed that same phenomenon where our wet season concentrations and our dry season concentrations were not so different. But again, we only had 2 or 3 real dry season samples. So again, not enough to draw that conclusion just yet. But again, we're gonna have that conversation, see what else, resources we have that we can allocate towards this question, and move forward going from there.
I think Tom has the mic now. The other Tom.
Thank you. Wanted to follow up on the storytelling theme, the 2 pieces, Dave, on the nutrient front about retrospective and prospective. We've talked a lot about the synthesis that Tom just touched on, but specifically from the historic about the need for a simple conceptual model to put out there, and your sense of what the pieces are, the key pieces that are still missing that we need to put effort into to get that, our best guess, best hypotheses about what caused the 2022 bloom, and as that's going to factor into my next part, the looking forward to feed into developing and where we are in, or you're thinking about the early warning system that we've been colloquial kind of talking about, integrating the satellite imaging and the telemetry and all the other real-time data. So conceptual model piece, the missing pieces, and then your thoughts on the early warning system.
Yeah. Well, going also back to Jim's point earlier, I completely agree that we need to be putting some substantial work towards, and we are putting substantial work towards the storytelling side. I also will readily admit that I feel like I find myself thinking often about if there's going to be a $10 billion potential decision made, it's not going to be like a 2-page glossy description is not going to carry the day. And so I think that's where we've placed much of our emphasis is on the more in-depth dives into these things. And to Tom, but the step from there to being able to make that information more usable, let's say, I'm looking forward to working with people on that, including people on our team, and as well as Sienna and others. And also look forward to people's advice on that, like what would be useful. In terms of, Tom, to your point about the conceptual model part, I think a lot of that is included in various iterations of documents, and we're actually actively working on those things as we speak. We do need to update that. Some of the things that, and this goes back, I think, to an earlier question about why were nitrogen concentrations higher in 2022 than other years? Was denitrification turned off? I actually think it could be the other way around, that there was a lot higher amounts of production and bioavailable carbon and denitrification in those other years where nitrogen was artificially low, which is a potentially concerning idea. One of the reasons for that could be lower suspended sediment concentrations, which could be a real game changer in terms of the bay's resistance to nutrients. So these are some of the things that at this point we can bring them in as hypotheses and competing conceptual models. We're still very much in the process of trying to put quantitative estimates and confidences around some of those. But I definitely agree with your point. On the remote sensing, you mentioned the early warning system. So we did get up and running with that web browser-based version of it, of being able to see chlorophyll on a daily basis, turbidity on a daily basis, as well as we now have the moored sensors coming in with telemeter data. I think the information is incredibly valuable for us to be able to understand, is something coming? We are using it to inform stakeholders. I keep coming back to, though, my thoughts keep coming back to the question of like an early warning system for what? Like an early warning system to inform the public is really, yes, that's really valuable. To inform the stakeholders and the regulators, really valuable. When, to Don's question, it's not like you're going to flip a switch on the discharges. That's a different type of early warning in terms, and like maybe even a different level of investment that you put towards the early warning system. And so, but yeah, I look forward to talking with you more about how far we might want to try to go with that.
Okay, we have 1 minute. We're going to take one more question, and there's a lot of whispering, so I feel like Martin was given some instructions to share. So let's get Richard's question in, I think, first. Does anybody have another one? I haven't seen any other hands go up. Oh, Ellen. Okay, can Ellen and Richard split their last minute? Can you ask a question in 10 seconds, get a 10-second answer, and then, yeah, okay. Because we know we want to get to the next phase of this day.
Okay, just real fast. So I just said, thank you, Dave. This has been absolutely fascinating, these slides. Just a very practical thing. So the process is you've got a stakeholder group that you're meeting with, the POTWs that are influencing and managing this, going towards this 40% reduction target. I mean, they're all influencing and contributing to the design and how you're working.
Collaborating. Yes.
Right, which means you tell them what to do or what.
No, all shared platform of information. Okay, good. Richard's question.
Okay, this is my third question of the day, but I've missed the last 2 annual meetings, so I'm still averaging 1 per annual meeting. So, David, thank you, and to your team, thank you for all of the amazing work you've done over the last decade. So it's really a monumental collection of effort and a body of work that you should really be proud of, so thank you. The question is about how comfortable you are and how you're hedging your bets about having heterosigma akashiwo as kind of like the most wanted, and I know that we've talked about this and you've thought about it. There's other organisms that have the potential to cause mischief, and not all of the work, in fact, is that the NMS is doing is dependent upon or assumes that heterosigma is going to be the bad actor, but some of it is. So I guess it's kind of like the question to you, and maybe Dan could weigh in. What have you been continuing to see in the data in terms of the species that are there that gives you more or less confidence that, at least for now, for some reason, it's heterosigma is going to be the most likely culprit for a problem, or not? So kind of like, how is the data influencing your thinking on that question?
I'll try to be really brief.
Mike. Pick up the mic.
So as we dig more into the data, we see that heterosigma is the time that bloom occurred was the time of year when it was most prevalent. And it's present about 60% of the time during that time of the year in that region. And so, and then there was 1 year where it decided to go rogue. And so there are other organisms, and Dan can speak to this, that have similar seasonal cycles, seasonal dynamics. I think in the terms, I definitely feel as though we should pursue with caution going too deeply into a heterosigma only way of thinking about things. I think there could be a lot of value to going into a more of a type of organism. So organisms that can swim and that tend to bloom during certain times of the year, and have a bit of a broader swath that we're looking at. And Dan, I don't know if you'd like to add anything more to that.
Yeah, I think I would totally corroborate that in the sense that heterosigma is one of the suspects we looked at, and we have a report on the way in review right now about investigating a kind of club among those 14 or so taxa that we're always on our suspects list based on worldwide reports of them being HABs. We know of a few of them that seem to correlate to each other in terms of their relative abundance in specific seasons, such as that August to September window. And we think that that relates back to their ability to swim, their ability, potentially their mixotrophy, their ability to scavenge other sources of nutrients. It could also contribute. And so we are trying ourselves to not become myopically focused on heterosigma only.
Richard, one last quick response to that point. There's even, I guess, another layer to this, which is the time of year that we might be focusing on. The permit is now a seasonal permit from May through September. Much of the time that we've spent working on these issues have been focused on spring blooms from, say, February through April. And either we were completely wrong before, and that was not at all a time to be looking at, and I don't think that that's the case. So in other words, like beyond just the organism, I would say that there's also time of the year that we should be thinking about in terms of critical conditions still, keeping those in view.
All right, that's it. Thank you. Thank you. So, no need to rush, as I said before, because so we could take your time to gather your stuff and perhaps have a little bit of interaction as you leave the building. But a couple things to, first of all, I'd like to thank everybody who participated today, their moderators and speakers. It was overall a great success, right? And again, thank you all for being here because you helped make it a success through your questions and interests. And we also want to have your input on how to continue to make things better. So this QR code is all about providing feedback on the annual meeting. I want to remind you that during Sierra's presentation, she briefly flashed another QR code to get input on communication, on the communication strategy. So at this point, we're going to do a follow-up. There'll be a follow-up email to all of you on a survey for the meeting today, but you can start doing on your own anyway, but you'll look forward for another one to tell Sierra how we want her to communicate to us and to others, right? So, and so what else? We're pretty much done, except again, we got half an hour before we have to vacate the building, but as soon as you want, I encourage you all to go across the street to the Jupiter. We'll carry on social interaction and have some fun dialogue. It's always an exciting group in dialogue that I've experienced over the years. So let's go have some fun and let's all keep up the good work, okay? Did I cover everything?
Thank you.